So hi everyone, welcome to tonight's webinar. We'll just give another couple of minutes just to let everyone join, and then we'll kick off in a couple of minutes. Okay, so I think we'll kick off anyway and we'll get started. So hi everyone, welcome to tonight's webinar on PGTA. My name is Katie and I work on the marketing and communications team here at SIMS. And tonight I'm joined by Aimee Walsh, and she is our lab manager down in our Cork clinic. So she'll be taking you through what is PGTA, who it might be suitable for, and all the ins and outs of it. So if at any point throughout the webinar you have any questions for Aimee about PGTA, what goes on in the lab, any of the process, please pop them into the chat and we'll answer them in our Q&A section at the end of the presentation. So first I'll just give you a quick introduction to SIMS. So SIMS is part of the Virtus Health family. Virtus Health is one of the world's leading fertility groups. So we share experience and knowledge from around the world, and this ensures our patients get access to the very best medical care and knowledge and expertise from different clinics and different partners in the Virtus Health family. So SIMS itself, we have 3 main clinics in Ireland. We have one in Clonskea in South Dublin, we have one in Swords in North Dublin, and we have one down in Cork. We also have our 3 satellite clinics, and they're located in Carlow, Limerick and Dundalk. So these are for patients who want to get their bloods and scans done throughout their cycle, so they don't have to travel to the main clinics all the time. So with that, I'll hand you over to Aimee and she will talk you through everything PGTA. So thanks Aimee for joining us tonight.
Thanks, Katie. And thank you everyone for tuning into this discussion tonight. My name is Aimee and I've just finished my stint as the acting lab manager. So I've just returned to my normal day job as a senior embryologist here at SIMS IVF in Cork. This is my 8th year as an embryologist, and I absolutely love this job. I'm from New Zealand originally, but I was trained as an embryologist in Sydney at IVF Australia, which is also a member of the Virtus Health Group. Here I specialised in PGT. That was my main lab task, about 4 to 5 days out of the week and also every second weekend. So I'd like to welcome you on behalf of the SIMS IVF team to our state-of-the-art embryology lab. And this evening we're going to talk about the IVF cycle from the perspective of the lab, and then delve a little deeper into my passion, Preimplantation Genetic Testing, PGT. So this is actually our Swords lab. They have a lovely shiny new lab up there. We're a little bit smaller down here in Cork, but we do welcome you to our lab here. So before we can do any testing on the embryos, we have to create them. So before we have the pleasure of meeting you in the lab, you'll go through the stimulation and monitoring phases of the cycle. The stimulation will cause multiple follicles to develop in the ovaries, rather than the usual 1 to 2 in a normal monthly cycle. Your progress will be tracked through a series of ultrasound scans and hormone blood tests. And when the time is right, you'll take your trigger injection, which prepares the eggs inside the follicles to mature and makes them easier for us to collect surgically. So down here you can see each follicle is punctured with a needle, and the fluid is suctioned out into these tubes here. These tubes are passed through a hatch window from theatre into the lab, where us as embryologists will transfer that fluid into dishes so that we can search for eggs under the microscope. Now, while we wish there was one nice egg in every follicle, this isn't always the case. Some follicles develop without eggs inside them, and some of them could be cysts. And, cysts look exactly the same on an ultrasound as a follicle. And not all follicles are going to respond the same way to the medication. So the egg might not release properly in order for us to collect it. But we will collect every precious egg we possibly can. So eggs come surrounded in a cloud of cells called the cumulus. We transfer these little clouds as we find them to a wash dish. And then when the egg collection is over, we transfer them all into a clean dish, like you see here, and we place them in the incubator for a few hours. Now down the bottom here, you can see what the egg looks like under the microscope. We've got the egg in the center here, surrounded by a jelly-like shell, which is called the zona pellucida, and then that cloud of cumulus cells on the outside. Now, while this is all happening, the male partner will be shown to a semen collection room, where he can produce his sample that will be used to inseminate the eggs that same afternoon. Alternatively, we could use previously frozen sperm from the male partner, or frozen donor sperm from one of the sperm banks that we work with, or surgically retrieved sperm, which we call a TZ. So in this video of a fresh sperm sample, when we get it into the lab, we're looking for several things: the concentration, or how many sperm there are, the motility, how well those sperm are moving, and the morphology, what they look like. So the majority of human sperm is actually abnormal. This little guy here on the left, that's what we're looking for. That's a normal sperm cell. And it's actually normal to have only about 5 to 8, sometimes a little bit more percent of normal sperm. So don't be alarmed if you see that your report says you only have, a low number of morphologically normal sperm. So we will prepare the sample to filter out the dead sperm and any debris, as well as the seminal fluid, so that what we're left with is just good quality moving sperm. Depending on the quality of the sperm sample, inseminating the eggs in the lab can be done one of 2 different ways. The first is what most people think of when they say IVF, meaning that we simply mix the eggs and sperm together in the dish and leave them overnight to do their thing for us in the incubator. This will be recommended if the sperm sample is good quality. Now, if there are fewer sperm than we'd like, or they're not moving very well, we'll perform what's called an ICSI insemination. So this means that a single sperm will be injected into each of the mature eggs we collect. This is common in cases of male factor infertility, or if a previous cycle using conventional IVF hasn't worked out the way that we'd like. So this is an example of the 2 different methods of fertilization. So IVF, or in vitro fertilization, literally means in glass, referring to those good old days when lab equipment was all made of glass instead of the plasticware that we use today. It's quite difficult to film because it's performed at low magnification using microscopes that don't have cameras attached to them. But this image is a good representation of how it actually works. So we add a calculated amount of sperm to the eggs, the dish with the eggs, and they swim their hardest to try and be the first one there. They're actually using signals coming from that cloud of cells, the cumulus, to guide them. This is the least invasive technique and allows the egg to play a role in selecting which sperm it wants to be the winner. Then we have ICSI. This stands for intracytoplasmic sperm injection. Now this video here, this is me catching a single good sperm. So I've tracked him down there, and we give the tail a bit of a swipe to stop him from moving. This also helps to release some activation factors, which will help with the fertilization, and also stops them from getting away from us. So we use suction to bring the sperm into the injection needle, and then go into a separate drop where we've got our egg. Now the cloud of cells has been removed so that we can look very closely at the egg and see if it's mature. We know that these eggs are mature because of this little fragment up the top here, that's the polar body. That shows us that the egg has gone through its final maturation and it's ready to meet the sperm. So we use some gentle suction to hold on to the egg. We don't want it going anywhere. And we make sure that we're perfectly lined up. We get the sperm to come right to the end of our needle. We check one more time that and then we go for it. So we pull back a little bit on the cytoplasm first. We want to make sure that this stretchy outer membrane, inner membrane will break. And then we release everything back into the egg, including the sperm, and that's it. Simple as that. So either way, the eggs will be left overnight in the incubators and checked on in the following morning. It's about 18 hours later that we hope to see those signs of fertilization. So ICSI is probably the second most difficult skill we learn as embryologists, and we go to great lengths to train at ICSI. We want to be as skillful and with as much precision as possible. Now, ICSI does come with a small risk that 5% of the eggs may not like being poked with a sharp glass needle, and they may not survive overnight. This is why we like to only do ICSI for those who require it. So other than a poor quality semen sample, this would be those surgically retrieved sperm that's taken directly from the testicles, or for patients who are using their frozen eggs, because we stripped away that cumulus cloud in order to freeze the eggs. So we wouldn't be able to do IVF once we thaw the eggs out because the sperm wouldn't know where to go. So we have to put them in directly. This video actually, I performed this ICSI last year. And this very egg and sperm went on to become a very cute little baby girl named Fia. She was born in April of this year. So the next morning, we're looking for those all-important signs of fertilization. And what we want to see here are these two little circles. This is what we call pronuclei. So one of these has come from the egg and the other from the sperm. So these two pronuclei show us that an egg is normally fertilized. Anything more or less is not right. You can see this one here has one large pronuclei, and this one here has three. So these images come from our time-lapse incubator called the Embryoscope, which is a camera inside and takes a picture of each developing embryo about once every 10 minutes. This allows us to monitor the embryos without taking them out of their nice warm environment, and allows us to capture images of the embryos, which we can show to you at the time of embryo transfer. So over the next 5 days, we're going to monitor the embryos, keeping an eye out for things like the number of cells, how evenly those cells are dividing, and how much fragmentation is happening. That is, how many little pieces are breaking off when one cell divides into two. The embryo should divide nicely from one cell into two, and two into four by the end of day 2 in the lab. So on day 3, we'll see those 4 cells become about 8 to 12. And on day 4, we might see up to about 16 individual cells, which we call blastomeres, before they all start to compact back together and form what's called the compact morula. This is the time for each cell to get in the perfect position depending on what part of the embryo it's eventually going to become. Tiny fluid-filled pockets will then start to appear as the embryo develops into the blastocyst stage. There are 2 early stages of blastocyst: cavitating or very early blastocyst, and early blastocyst, before the embryo becomes full-sized and is ready to either be transferred fresh or frozen for later use. We can now clearly see that there are 2 different cell types developing in the embryo. We've got the ICM, or the inner cell mass, and that goes on to form the baby for us, and the trophectoderm around the outside, which goes on to form the placenta and the amniotic membranes. On days 5 and 6, we also see expanded blastocysts that are growing and stretching, pushing outwards from the inside on their jelly-like shell until the shell is so thin that pressure from the inside causes it to split open, and the trophectoderm can continue to push its way out. This might be a little bit, a lot, or the embryo might come all the way out. You can see the shell left there in the background. The shell has protected the embryo during its early life, but it can't implant into the lining of the uterus until it hatches all the way out. It's fairly rare to see fully hatched embryos in the lab. This tends to happen naturally around day 7 or 8 of development. So that's after we've already placed the embryo back in the uterus. As well as noting the developmental stage that the embryos reach, we're also going to grade them for you. Our grading system features a number followed by 2 letters. The number will be the stage of development. Then the first letter will be the grade for the inner cell mass. That's going to be either A, B, or C, with A being the best and C being not suitable for use. The second letter is the grade of the trophectoderm. We also grade this A, B, and C, just like we do with the inner cell mass. These grades help us to choose between embryos, ranking them from the best looking one down, making sure that we give you the best chance of success possible with each embryo that we transfer. So before we move on to exactly what is PGT and what does it involve, let's take a peek at 4 embryos that we looked after in the Cork lab recently. So these were fertilized by ICSI. We see the 2 pronuclei appear, and then we watch as they divide, all developing at their own pace. Continue to watch them, leaving them completely undisturbed until they become blastocysts. 3 hatching blastocysts, to be specific, and a full blastocyst over here. So watching the cells divide as the embryo develops and ticking off each milestone as it makes it can only tell us so much. We can't see what's going on inside the cells. Are these embryos genetically normal? Do they have a higher chance of leading to a live birth, or do they not have what it takes to implant? In a non-PGT cycle, we transfer the embryos back one at a time, starting with the best looking one. So we'd transfer this one first with an AA grade. This would be our second choice, an AB. Third choice is another AB, followed by this guy who is a BB. But what if this isn't the right order to maximize the chances of success? And this is where PGT-A comes in. So each embryo in a PGT-A cycle will be artificially hatched on day 4 of development. This is where we use a laser to create this small hole in the embryo shell so that as it grows and expands, the trophectoderm can begin to stick out through this hole. This allows us to take a small sample of these cells without needing to go inside the embryo, and we can stay well away from the inner cell mass. Now that we know how embryos are created and cultured in the lab, we can talk a little bit about the genetics and why this is important in a successful pregnancy. So every single cell in our body contains a nucleus, which is essentially the brain of the cell. It houses all the instructions that cells need to carry out their function. Whether this is a beating muscle cell in the heart, a granulosa cell providing nutrients to the egg and guiding sperm to its location, or this is Sertoli cells within the testicle responsible for supporting the developing sperm. The reason each of them know what job to do is due to the specific set of instructions inside their nucleus. These instructions are housed within the chromosomes. And the cool thing is that every cell in our body has the exact same set of instructions as every other cell, no matter what type of cell they've become. So as humans, each of our cells contains a set of 23 pairs of chromosomes. 1 chromosome in each pair will be inherited from your mum and the matching one from your dad, for a total of 46 chromosomes. These chromosomes are made up of coils of DNA, and the DNA is what provides the code needed for all living things. Genes are specific little segments of this DNA, which are responsible for, among many, many things, the physical characteristics that make us unique. If DNA were the ingredients, genes are like the recipe, and the chromosome is the particular chapter of the recipe book that is the nucleus. The information that genes carry makes you who you are and determines what you look like. Sometimes it's simple things like straight hair or curly hair, blue eyes or brown, or more complex traits like how well you handle stress or how emotional you get during a sad movie. They can also be responsible for passing on some not so nice things, such as diseases, sickle cell anemia, cystic fibrosis, some cancers, or Huntington's disease, for example. So our body has a complete instruction manual of 46 chromosomes that tell our cells how to behave. If one of these chromosomes is missing, or there's an extra one, the body functions differently, or may not even be compatible with life. So this is a normal karyotype, or a chromosome picture. There are 2 of each of the autosomes, which is the first 22 pairs, and then depending on your gender, either an X and Y sex chromosome or 2 X's if you're female. So you'll have your karyotype checked before you begin your PGT cycle. So what is PGT and what are we looking for? You might have seen or heard the terms PGS, PGT. These are old names for what we now call PGT, Preimplantation Genetic Testing. As the name suggests, we're testing the genetic makeup before they're implanted into the uterus. This differs from prenatal testing like the NIP test, as that happens after the pregnancy is established and the fetus is developing in the uterus. PGT in a nutshell is a test performed during your IVF cycle, with the goal being to improve your chances of a successful pregnancy and having a healthy baby. PGT-A tests for the number of chromosomes in the cell. As we've discussed, humans should have 23 pairs of chromosomes for a total of 46. Embryos with the incorrect number of chromosomes, called aneuploidy, typically do not result in a successful pregnancy, but some may lead to the birth of a child with a genetic condition, such as Down syndrome, where the baby has inherited 3 chromosome 21s instead of just 2. Embryos with the correct number of chromosomes have a better chance of leading to a successful pregnancy. PGTA identifies these embryos so that we can select the embryo with the best chance of leading to IVF success and a healthy baby for you. PGT-M is used by people who have a known increased risk of passing on a genetic condition caused by a single gene called a monogenic disease. For example, patients who have cystic fibrosis or Huntington's disease and don't want to pass these diseases down to their children. It's important to note here that this is not a screening test for a variety of monogenic conditions. The test is specifically tailored to the individual patient's condition, as 2 people with cystic fibrosis might have very slight differences in the mutation that's causing the disease in them. And PGT needs to be very specific to the individual in order to be accurate. Lastly, we have PGT-SR, which tests for structural rearrangements in the chromosomes. This is where the total amount of genetic material is correct, but part of one chromosome is broken off and switched places with a piece on a different chromosome. People with these chromosomal rearrangements are at an increased risk of producing embryos with the incorrect amount of genetic material, as these mismatched chromosomes interfere with the normal way that DNA replicates itself during cell division. Chromosome rearrangements are rare, and a person born with them may be completely normal or may have some degree of disability depending on where the break in the chromosome is located and where the other part of the chromosome has attached. So we'll focus on PGTA from here on out, as this is 99.9% of the PGT we do here at SIMS. The PGT cycle at SIMS Corp is a partnership between the patients, us, and a company called Cooper Genomics, who perform the genetic testing for us. So we've got the eggs and the sperm, and we've created and hatched our beautiful blastocysts. It's time to take the biopsy sample and send it off for testing. So how do we do a blastocyst stage biopsy? First, we check the embryos on the morning of day 5 and grade them to see which ones are ready now and which ones might need that extra day and will be rechecked on day 6. When grading, we have to keep in mind that the embryos, we're going to be taking some cells away, and we need to be confident that what we leave behind is still a good quality embryo that's capable of surviving the freezing and thawing processes. So watch biopsy here. We hold the embryo securely in place on the left here, and again, making sure that we're perfectly in line, we apply gentle suction on this biopsy pipette and slowly draw a few cells up into the pipette. We have to be quite gentle as the cells of the embryo are fairly fragile. It looks brutal, it looks like I'm sort of tugging and stretching and bouncing on this embryo, but what I'm trying to get it to do, I'm trying to encourage it to collapse. That's going to make just make things a little bit easier and make those cells slide into the pipette a little bit smoother. So with the laser here, I can use to let some of that pressure out. You can see the cells moving in quite a bit easier now. So we keep that suction until we have a few cells drawn into the pipette. We're taking a small sample of the vitrification, so we're not going anywhere near the inner cell mass down here. This part, the cells that we're taking are not part of what's going to become the baby. So we're not doing the embryo any harm in any way here. So once we've got the cells we want, we use the laser again just to loosen the connections between each cell. Once we're happy that they are nice and loose, we're going to do something that's called flicking. But we need to completely detach the cell sample from the embryo. So we'll zoom out a bit so we can see what we're doing. We're making sure, again, we're always perfectly aligned and that we're happy with the amount of pressure we're putting on the embryo. Put them together, and quick as that, we flick the cells apart. So it looks a bit scary, but this is the most gentle way of doing the biopsy. There were some older methods where you would keep pulling and pulling and stretching until the cells broke off on their own, but that causes damage to surrounding cells. Or using the laser to completely separate the cells from the biopsy from the embryo. But that causes an intense spike in temperature at the point of the laser, which will degrade the DNA in the cells. You don't want to do that. So it looks scary, and believe me, the first few times we do this on, well, the first many times we do this on training embryos, it's terrifying. You end up holding your breath, but it's a controlled movement, and the embryos survive much better this way than compared to the older ways of doing it. So we release the cell sample that we've got, and it can be a little bit sticky sometimes. We check on our embryo to make sure it's happy, and then we check on our cell sample to make sure that's whole and intact there. So this is, like I said, this is the best way to get good quality DNA, and good quality DNA is how we get good, clear genetic results. We only have senior embryologists at SIMS perform embryo biopsies, who already have years of experience with delicate lab procedures like ICSI. Our team are very highly skilled at this. I myself have biopsied over 2,000 embryos at this point, and you know, we would only do this. We would only move on to working with patient embryos when we were completely satisfied that we have got this technique down to a fine art. So once we've prepared, once we've got the biopsy sample, we take the embryo to a separate dish to wait for freezing. And we must prepare and package the biopsy sample to be sent to Cooper Genomics. So to do this, we cover up our scrubs with a sterile surgical gown, a face mask, and some long sterile gloves, just like you've seen surgeons put on in movies. We do this so that we don't contaminate the biopsy samples with our own DNA. You are wanting to know the genetic makeup of your embryos, not your embryologist. So we carefully load these samples into tiny tubes. They are just 2 centimeters long, and we're putting the cell sample into an even tinier drop of a special media. It's just 2 microliters, so 0.0002 of a milliliter. Once the cells are inside that little drop, we can put them in the freezer and ship them to Cooper Genomics lab in London for analysis. So the PGT results are going to take a few weeks to come back, so we need to freeze the embryos while we wait. Now we say freeze, but what we actually do to the blastocysts is vitrify them. The vitrify literally means like glass. If you think about glass, it can be a gooey liquid state, but if it's cooled down really fast, it becomes solid and brittle, and it can be returned to that gooey state by simply reversing the process that cooled it down in the first place, like by heating it up. So to successfully freeze embryos, we actually have to dehydrate them because our cells are made up mostly of water. And when you freeze water, it does two things. So if you've ever put a bottle of drink in the freezer to cool it down quicker and then forgotten about it and come back to an exploded mess in the freezer, it's because the water inside the drink bottle expanded when it froze. And if you've ever had some frozen fruit, taken it out and defrosted it, it's always a little bit soggy and a bit mushy compared to fresh fruit. That's because the water inside the cells formed crystals when it froze, and those sharp crystals damaged the walls of the cells. So we dehydrate them and remove some of the water, place them in a drop of cryoprotectant media before loading them onto these devices called high security straws. These skinny straw-like devices have an insert inside them, which holds the embryo in place so we can seal each end before quickly plunging it into liquid nitrogen. Liquid nitrogen is minus 196 degrees Celsius and cools the embryo so fast that it passes directly from the liquid phase to the glass phase before it has any time to form these nasty ice crystals. The embryo is now essentially frozen in time, or vitrified in time, and will not develop or degrade in any way while they're stored at this temperature. So this is a photo of a tank containing many patient embryos. Each patient is given an address within the duo so that we know where every single embryo is at any point in time. For example, your embryos may be stored in 6, 5, 20, which means duo number 6, canister 5, canisters being these buckets here, and cane number 20, these metal rods, which are allocated to a single patient. Now, embryo freezing has come a long way in the last few decades, with the current embryo survival rate, meaning the embryos coming out of the freezer exactly how they went in, from vitrified embryos being over 98% in this, using this technique. Now, speaking of frozen embryos, it is possible for us to perform PGTA on embryos that have already been frozen. In this case, we thawed out 4 day 5 embryos. You can see the embryos start off a little collapsed here and crumpled looking. That's perfectly normal. And this is the result of the dehydration they went through during vitrification. We use the laser to create those little holes, and we pop them in culture for a few hours to recover. They will return to their original shape and size and start to push out some of their trophectoderm cells for us to biopsy. Most embryos will be ready to biopsy the same day that they're thawed. However, a small number of embryos won't be ready, and we'll keep them overnight with the hope of biopsying them the next day on day 6, like this right here. So thaw biopsy and refreezing of embryos is only recommended when several embryo transfers have failed and all other factors potentially affecting implantation have been explored. While this is a semi-routine process, the added strain put on the embryos during a thaw biopsy and refreezing can potentially result in a negative impact on the embryo's overall quality and an increased risk of the embryo not surviving, as it's going to need to undergo yet another thaw when it comes time to do the embryo transfer. So we've partnered with Cooper Genomics to perform the genetic testing aspect of the PGTA cycle. Cooper Genomics have over 27 years of experience with PGT testing and have tested over 500,000 samples at this point. So we've created our embryos, we've biopsied them, and now have them safely stored away in liquid nitrogen. The biopsy samples undergo a process called next generation sequencing, or NGS. First, a machine at Cooper Genomics in the Cooper Genomics lab copies the DNA inside each of the cells hundreds of thousands of times. This makes the information much easier to interpret now that there's more of it. The NGS process produces results like this, which are analysed using AI and the expertise of senior lab staff at Cooper Genomics. You can see these little dark green dots are all lined up along this light green line at 2 mark, meaning that there are 2 copies of each of the chromosomes here. You can see the chromosomes are numbered down the bottom here. If we look at the top section, this thick grey section here is for the X chromosome. And we can see that this person has 2 copies and is therefore a female. So a normal female karyotype is represented as 46XX. And if we look down here, this person has 1 copy of the X chromosome and 1 copy of the Y. So this person is male and their karyotype is 46XY. So these 2 karyotypes are what is known as euploid, meaning the correct number of chromosomes are present and accounted for. Now these ones here, this is what we don't want to see. This top one here still has a chromosome count of 46. However, we can see that there's less DNA than there should be here at chromosome 7, known as monosomy, so only 1 copy. And there's more DNA than expected here at the 8th chromosome, known as trisomy. This embryo has 2 X chromosomes, so this is a female, and their karyotype would be 46XX minus 7 plus 8. This bottom one here has an extra copy of chromosome 16, so it has a total of 47 chromosomes altogether. And again, as a female, so XX, and plus 16. So these embryos are abnormal or aneuploid. So these aneuploidies are not compatible with life, what we call fatal aneuploidies. So if they did somehow manage to implant, they're more likely to result in an early miscarriage. So we've done the biopsies and they've processed the samples, but they're not going to present the results in the graph form like that to you. So now it's time for them to provide a report of their findings. This is an example of how the results are reported. The samples are numbered according to the embryo number that we assigned all the way back when the eggs were collected. The embryo keeps this number throughout so that it's always clear. The frozen embryos are labeled with these scannable barcodes. These barcodes work with our electronic witnessing system and always ensure that we know exactly what embryo we're thawing and what the PGT result is. So let's go through the type of results you might see on your report. First and most desirable is the euploid. So the embryo has that correct number of chromosomes in every cell. These embryos have a higher chance of pregnancy, a lower risk of miscarriage, and a reduced chance of a baby being born with a genetic condition. Another possible result is aneuploid, which means incorrect number of chromosomes. And this could simply be 1 or 2 errors, like this one here, or it could be 3 or more, which would be a complex abnormal embryo. Aneuploid embryos, because they don't typically result in a pregnancy, or if they do, they have an increased risk of ending in a miscarriage. They're deemed not suitable for embryo transfer, and we remove these from cryostorage. Now, there are a number of aneuploidies that are compatible with a live birth. These are the trisomies that result in Edwards, Patau, or Down syndromes, or extra or missing sex chromosomes like these ones here. So because they could potentially result in a live birth, we'll keep these embryos in storage unless you give us consent to remove them. Next possible result option is mosaic. Now this is where things get a little complicated. Mosaic embryos contain 2 or more different chromosome patterns within the one embryo, so a mixture of both normal and abnormal cells. So if we look down here, we've got our normal euploid embryo, where the normal cells are green, and over here is a fully euploid embryo with all blue cells, and the mosaic embryo with a bit of a mixture there. And this is the site where we're going to be taking the biopsy from. So the mosaic embryo with the mixture of cells. Now this could be reported as a low-level mosaic, meaning that 20 to 40% are these blue abnormal cells, or a high-level mosaic, meaning more than 40 and up to 80% of the cells in the biopsy are abnormal. So it may be an option to transfer these embryos based on whether it's a high or a low level of mosaicism and what chromosomes are affected. but this must be discussed with one of our doctors and possibly even a qualified genetic counsellor at Cooper Genomics. The last option no one wants to see is no result. This can be one of two different types. The first is no intact DNA, where the initial step where the machine was making those hundreds of thousands of copies, that process has failed. Or we might see quality insufficient for interpretation, where the DNA was able to have those copies made, but the result was not clear enough to pass the strict quality control standards that Cooper Genomics have in place to ensure that they're only providing you with the correct result. So both of these can happen due to poor quality samples. The good thing is that it's very rare. In our first year of PGTA here in Cork, we biopsied about 200 embryos and had a no result come back for 4 of them, which is 1.9%. So that's far below the 5% rate, which is considered acceptable. There's a number of options available if you do get a no result, which you can discuss with your doctor. You could choose to have us attempt a rebiopsy of the embryo, and that would be in a thaw biopsy refreeze, like we saw with the frozen embryos. You could transfer the embryo without knowing the genetics, just like you would in a regular IVF cycle without PGT. Or if you feel more comfortable, you could always ask us to discard that embryo for you. So we've done the biopsies, the samples have been analysed and the report's been made. It's now time to meet with the doctor, discuss your results and make a plan for the embryo transfer, and hopefully get pregnant. So one of the most common questions we get asked when we're giving patients the final lab update is, does the grade of the embryo tell us anything about the genetics? Are better quality embryos more likely to be euploid? And the short answer is no, but we wish it did. So if we go back to these 4 embryos from earlier, we said that without PGTA, we would rank the embryos based on their quality, their grading, and we would be transferring this guy here first. But in this particular case, the PGT results actually came back with a completely different story. It turns out these embryos we would have transferred here first are both abnormal. They both have chromosome errors within them. This one here, particularly this embryo we would have transferred first, has that trisomy 16, the extra copy of chromosome 16, which is a very common cause of early first trimester miscarriages. So they're no good. And the embryo we really should be putting back is number 3 here. So this result came back normal, euploid. It's the most likely of the 4 to be a success. And this embryo here was a low-level mosaic. All these numbers and letters is just a complicated way of saying only a small piece of chromosome 12 is missing, and it's only in 20 to 40% of the cells. So transfer this guy first, and then if that one was not successful, or maybe a couple of years after the baby is born, we could potentially transfer this one. To give you some idea of the chances of having a normal or euploid embryo, it's usually determined by female age. I know we don't like to think about ourselves getting older, but this graph shows that the purple line is the percentage of normal embryos, and the red is the abnormal embryos. So if you're female and you're under 37, the chances of having a normal embryo is about 50%. And this drops quite dramatically to about 15% when you're over 42. Another really clear way of showing this effect of female age in our own Cork patients is we color code the embryos when the results come in. So like a traffic light system, green is good. That's our euploid embryos. Red is bad. Those are our aneuploid embryos. And orange embryos are those that require a little bit of thought, like the mosaics and the no results. So this group on the top here, these are our under 37s. The youngest patient here on the left, going to the older one on the right here. This middle group is our 37 to 40-year-olds, and the bottom group is our over 40s. So you can see a very clear increase in the amount of red and decrease in the amount of green as we go right the way through from a 31, almost 32-year-old to our oldest patient last year, which was almost 46. So who's PGTA for? Your doctor might suggest adding PGTA to your cycle if you're above the age of 38. We call this advanced maternal age. You can tell a man came up with that phrase. We've seen in those last two slides that you're more likely to have an aneuploid embryo if you're above this age. If you've already had a pregnancy that ended because of a known chromosomal abnormality, if you've suffered multiple miscarriages or had multiple embryo transfers that haven't worked and there's no other explanation for it. Some patients will opt for PGTA so that they can have more confidence in just having one embryo transferred and reduce the risk of having twins that's present when we transfer two. Or they may just want to have peace of mind. There's very few things that you can control during an IVF cycle. So some people like to know that they've done something tangible to increase their chances of taking that baby home. So what this really all comes down to is live births. Data from our partners at Cooper Genomics has shown that PGTA can increase the chances of having a successful pregnancy by only transferring those embryos that have what it takes to be a healthy baby. It can reduce the risk of miscarriage by avoiding embryos that were destined to fail. And this last one here, which is often overlooked, PGTA can result in a reduction in the potential emotional, physical, and financial impact of round after round of failed cycles. And this can also mean that the time taken to get that live birth from when you first walk in the doors is significantly reduced, allowing for the possibility of siblings. So why don't we use PGTA for everyone then? Well, you've seen that it's a very invasive technique. We don't improve the embryos by taking cells away from them. And there's always that small chance that the embryo might not survive the biopsy, the freeze, and the thawing procedures. So it's usually recommended when there's a potential benefit that outweighs the risks of embryos. So the other great thing that's really clear to see from Cooper Surgical's Cooper Genomics data is that PGTA is able to reduce the effects that maternal age has on success rates. You can see the non-PGTA cycles in the blue bars here, and the PGTA cycles in green. And we look where this pink line is here, we can see that a woman who is up to 42 and does PGTA on her embryos and transfers a tested euploid embryo has the same chance, if not more, of a lady who is 34 and is using a non-tested embryo. So thank you for joining us here tonight. I hope you enjoyed this webinar and I hope it's answered some questions you might have had about PGTA. I'll leave you with these here. This is genuinely us each day, loving the job that we do and the difference we get to make. And we do treat your precious blastocysts nicely, I promise. So I'll hand it back to Katie, and if you have any questions, you can feel free to get in touch.
Yeah, thanks so much, Aimee. That was super. It was really, really informative. It was really great to go through all those different sections and give everyone a bit of an insight as well to what goes on behind the scenes in the lab as well. So just before we go into the Q&A as well, we are offering everyone on the webinar as well a half price initial consult. So I will email everyone tomorrow with a code that if anyone wants to have a consult and have a chat with their doctor about PGTA or any fertility questions, that you have that opportunity as well. So thank you everyone for joining us as well. And I'll email everyone tomorrow with that code. I'll pop it into the chat as well and you can pop that into the new patient form if anyone wants a half-price consult as well. So there was a few questions that came in, Aimee, while you're giving the presentation. So one of the questions that we had in was, what is the current waiting time to get the PGTA results back?
So it's roughly 4 to 5 weeks after your egg collection. We ship the cells every 2 weeks to avoid. It's easier for us if we can batch them all together, and it's better for the cells themselves to go with more ice, more cold, more temperature to London. So we will try to book you in for your results call about 2.5 to 3 weeks after we actually do the biopsies. The results actually come in like a little bit before that, but we want to make sure that they actually are in before you see the doctor. We don't want to have to call you the day before and reschedule because the results haven't come in.
Yeah, no, that's great. Another question that we had, Aimee, which is quite interesting as well, is there anything you can do that can influence whether your embryo will be normal or abnormal? Is it down to sperm quality or egg quality, or is it purely genetics?
I wish there was something that we could do to improve things like this, or to show us in some way that, down the microscope, that this is the embryo, this is the euploid one that's going to work. There's really only evidence of reducing DNA damage, but that's not really aneuploidy. That's more damage to the cells, the DNA within the cells as they try to develop, and we can reduce that by quite simple things like getting enough sleep, taking general care of yourself, all of that sort of thing.
Yeah. Just check, there's another question that has come in, is to do with as well the biopsy process. So there was a question about how does the embryo replace the cells which are biopsied? So I believe this comes from the part of the cell that will eventually be the placenta.
That's right.
Is that correct? Yeah.
Yep, so we're doing that at the stage where the embryo has about 100 to 150 total cells. The embryo still has a few more days of development before it's hatching out of that shell, and then a couple more even before it starts implantation. And that whole time it's dividing, each cell is splitting into two, and it keeps going until there are over 1,000 cells. So it definitely can do without 5 to 8 of them. We used to biopsy embryos back in the day when they were at the 6 cell stage. So we were taking out one of those blastomere cells, and that's 1/6 of the whole embryo, and the embryo does perfectly fine.
Yeah, definitely. I think sometimes there can be a bit of confusion. People might think it's coming from the baby part, and that can, I think that's good definitely to that people understand that as well, where they come from. So that's great. So we have another question as well, just after popping in now, Aimee. So if the results come back as non-conclusive, does that suggest that there's a higher chance of something being wrong?
No, actually, Cooper genomics data has shown that when we rebiopsy the embryos and we send that second cell, the embryo has just as much of a chance of being normal, of being euploid, as an embryo that's only had one biopsy. Sometimes it's just the conditions of the biopsy. It can be very hard to pinpoint the exact reason why we don't get a clear result, and that's why we set the upper limit of 5%. We try to stay under 5%, but like I said, we're well below that 1.9%.
Yeah. So do we have another question here? Are the tested cells identical to the embryo cells?
Yeah, so there's no guarantee. PGT, it's a very accurate test, but we are taking that biopsy sample from the part of the embryo that doesn't go on to be the baby. But what people have done long before my time, before I got into this, they would take surplus embryos that patients had donated after their family was done. And what they would do is do multiple biopsies of the trifectoderm, and then also do some biopsies of the inner cell mass as well, and test those to make sure that the results we get from the trifectoderm are truly representing the inner cell mass. And it varies from method to method and location to location, but it's up above sort of 80, 85, 90%.
Great, thanks, Aimee. I'll just have another look if there's any more questions that have popped in. Is there any reason why it might not be recommended for someone?
We wouldn't recommend it if you're very young. Obviously, it's still your choice. If you want to opt into the service and you want to pay for it, that's your decision. But if you're in your early 30s and you've never been pregnant before, maybe you've been trying for just over a year or something like that, you're much better off just going for those fresh embryo transfers, because chances are most of your embryos are going to be normal anyway. We do also have to get those embryos to that hatching stage. Some patients, some older patients particularly, the embryos won't develop that far. We could still freeze them if they reach that stage 3, the full blastocyst stage, but that's too small and too early for us to biopsy. So we wouldn't recommend trying to push the embryos further than they're capable of going just to get that result, because we have to be confident that what we're leaving behind is still a good embryo. There's no point biopsying the embryo and getting a result if we haven't left enough embryo behind to actually implant.
Yeah, that's great, Aimee. It's all really informative. We're just nearly at time there, so I'll just have one last check through the questions. There's been some queries just around the costing. Do you know the costing of a PGTA cycle?
I don't off the top of my head, I'm afraid.
Yeah, we have the prices, I think, listed on the website as well. So if anyone is curious, if you go to the About section of our website, the up-to-date pricing is listed there, and it will give you an indication as to how much the PGTA cycles cost. So if I'll just have one more check if there's any more questions in. Thank you very much, Aimee, as well, for going through all that. It was great to hear everything firsthand and get a really good insight behind the scenes and see the videos. It's great. So I think that is all the questions we have in at the moment, so we can end the webinar there. Everyone, if you want to use the code, I've put it into the chat for anyone who would like to avail of a half-price consult, and I will also send an email tomorrow with the code for anyone maybe that's popped off or has missed it as well. So thank you very much, everyone, for joining, and I hope you all got something out of it and learned a lot about PGTA. And if you have any questions or queries, please feel free to contact us at communications@sims or through the website. One of our team members will be in touch anyway to discuss. So thank you, everyone. Thanks, Aimee.
Thanks.
Bye.